Hey everyone, good afternoon. I hope you guys had a good lunch. So thanks, Dr. Murthy, for a quick introduction. Yes, I think that was a tagline on my LinkedIn. So basically, my role has been both in the bioinformatics domain as well as AI slash machine learning domain to kind of connect multiple omics technologies, their results, such as genomic, proteomic, with other modalities such as radiology, which they call radiomic, and slide-based histopathology, slide-based imaging techniques. So the idea is how we could bring these data sets together to kind of correlate, let's say, between the radiology images, their image features, with the genomic features, which could be the genomic mutations, expressions, all those key factors which are reported for a patient, based on which it dictates the diseases such as cancer, and then based on these reports, these patients get treated. So when a patient enters a clinic, it goes through a lot of tests, right? MRI, CT scans, tumor biopsy, histopathology, slide imaging to evaluate the size, shape of tumor. And then the tumor gets scraped off, the DNA, RNA gets extracted, and it gets sent to the genomics laboratory for DNA, RNA quantification, expression, mutation analysis, etc. And then based on that, the therapies are given, then the patient therapy outcomes are received, and at some point the disease recur as well. So I'll try to encompass all those things in my talk today. Mainly the talk details tailoring imaging phenotypes to measure omic profiles for tumors using Imagine. Imagine is one of the software that I made through my PhD at Queensland University of Technology in Australia while I was still working at Dartmouth-Hitchcock Medical Center. I'm still working at Dartmouth-Hitchcock Medical Center as Assistant Director of Genome Informatics here in the pathology department. But my PhD was in a much more broader domain where it was called radiogenomics in the beginning, like connecting radiology data with genomics. But then it expanded to omic data sets, as Dr. Murthy alluded, proteomics, genomic, therapy outcomes, and disease recurrence follow-up matrices. I do have a close collaboration of my I did have the close collaboration of my PI with Stanford and the Indian Institute of Technology, Delhi. And currently I work at Dartmouth Hitchcock and kind of try to implement these techniques here so that I could bring it to the clinical workbench. So just a quick introduction of radiogenomics. Basically, as I said, a patient enters the clinic and goes through several different modalities that you could see here. So first, a brain image scan, for example, for a brain tumor gets taken place. Tumor ROI is marked by radiologist, based on which the tumor biopsy takes place, and then the tissue slides are extracted. The DNA, RNA are extracted from these tissues. From the stained tissues, stain markers are also analyzed versus the mutations and expressions. These genes are clustered into the biological pathways. On the upper end, you would see that what radiology scientists do is they also extract imaging features, texture, intensity, spatial, volumetric of that particular section of the tumor, and try to see if they could directly correlate this with patient outcomes. What radiogenomic field does is it merges these two features together to try correlate them through statistical methods such as Pearson and correlation and Spearman-based correlations, and then feeds it into machine learning models, variety of machine learning models these days, classification as well as regression-based models, to see whether the imaging profiles or the imaging features could dictate the genomic profiles of those tumors or vice versa. And this could basically encompass to any omic modality that I have expanded this study further to kind of accommodate that and try to evaluate the associations. And this could be done with respect to any cancer type that you could think of, because I mean, whether it's brain, lung, breast tumors, The same technique is followed, has been followed through years, but now is the time that we should connect these data sets together and try to figure out if we could directly predict some expressions, some gene or protein expressions, I would say, which are more effective from these image features, to hopefully improve the biopsies, to make them more accurate with a feedback mechanism, or kind of predict patient outcomes based on the images, which would be the most non-invasive way to treat cancer, and a good turnaround time in near future. But again, these all processes would need a very hardcore validation. And coming from a clinical lab, performing clinical informatics for almost 12 years now, there's a very rigorous requirement to get the research omic validated, and that's why the description of my profile says that I'm trying to balance the gap between research and clinical omics, such that all these research techniques could be validated and brought to clinical omic reporting for patient. So of course, with these kind of analysis, many laboratories these days, they do it through machine learning model coding in Python, and different platforms like Kaggle that allow to write loose codes that may get lost through the laboratory workflow, right? But there has been some existing tools for imaging omic studies, such as one from my co-PI earlier, Amaretto and Imaging Amaretto, which takes the imaging features and the genomic expression pathway information, but it lacks a very sophisticated user-friendly software, and also doesn't allude too much as in how these models play role, and their matrices are not being alluded much into a clear format. So we are speaking in terms of transparent algorithms, where even if you're using a machine learning model like linear regression, you want to know how your features are being treated, what are their RMSE and AUC scores, R square at feature level, like if you add or decrease the features, how would the model perform, and how would these scores get defined for the model, and then you get feature level importances thereof with the respective p-values. There was other software called Musa, which basically lacks the machine learning AI module, but has other sophisticated statistical methods like correlation heat maps and PCA. So the idea was to come up with a software that would basically do top three things. One, analyze the data sets with enough checks and balances built within, with enough automation in place so that you could feed in like imaging feature file with the omic feature file, and it would do the data normalization, it would do the QC, it would run models based on multiple parameters that you could provide very extensively with the click of buttons, either in a GUI format, or if you are informatics savvy, or command line version of it. And is a operational report allowing users to track their features very closely through the entire workflow, like which features are being correlated versus which are being thrown out. And then if you want to take all the features through the machine learning model, how are they, what are the correlation weights at each phase? And yeah, how do their importances get defined and get selected further for retraining? So ImaGene is a software that allows you to perform that. You can see on the left, it has a simplistic schema, whereas there's a more detailed informatic workflow on the right-hand side. As you could see, it accepts any two features files that you want to correlate and make associations and predict one data from the other. Remember that this could be any feature file, omic feature file that you wanted to use. You could even mix the imaging features with omic features in this file on the left-hand side and predict patient therapy outcomes. So we have had some studies done now. There's a paper out already there, which should be on my profile on LinkedIn, that basically uses two use cases, one in invasive breast carcinoma and head and neck squamous cell carcinoma, which we have depicted that using very highly qualified imaging features, which are curated by radiologists, they depict gene expression features for certain genes at very high AUC thresholds, even up to even greater than 0.9 in many cases. Initially, this had only like 3 to 4, regression-based models, but the scope increased with the development of imaging since past 3 years. So now we also have like classification-based models like random forest, MLP classifiers, support vector machines, etc. And as you could see, it goes through a lot of feature selection, feature scoring, grid search, and where you could do significance tests and validate the data set after training the model and get nice reports out into the place which would depict these profiles. I had a demo, but I think it would kill the time. But you could easily go to my paper and sort of scroll through the ImaGene's GitHub page, which would basically allow you to see the demo through its wiki. So I'll straight away go to a use case in the case study that we studied using ImaGene. So the first case study was the invasive breast carcinoma. And what I'll do is I'll just stick with the topic and go through both the omic domains, genomic and proteomic here, right? So invasiveness in breast carcinoma is defined by a tumor starting at the lobule or duct phase and then spreading out further into the breast tissue. And as you could see, these are measured initially through the radiology scans, mainly MRIs or ultrasounds. But here the modality that we used was MRIs, and basically you could characterize these tumors such that they could be segmented and could have many imaging features associated with it, like texture, intensity, gray level matrices, etc., that gets extracted using many tools these days in computational domains such as radiomics or other radiomic tools developed by universities like Stanford. And then that gives you a lot of imaging features which could then be correlated with the genomic expressions obtained from these tissues that get biopsied. And that would also speak to protein expressions and obtained from different arrays such as RPPA. Here is the result from the very latest paper. You could just blast it through PubMed, and I don't have a PubMed ID, but again, my profile has this paper where basically we tested a lot of model types such as decision tree, multitask, elastic net, regression, multitask lasso, all of them very flexibly operated through ImaGene on rather a cloud-based system with a very quick turnaround time to match up with the gene expressions at various thresholds that you could see. These thresholds were normalized. These are like fragment per kilo megabase thresholds for the expression, for the RNA expression dataset, RNA-seq dataset, and these intervals indicate low to high expressions at which these genes got predicted using this model type, using the respective imaging features. And you can see that we were able to achieve high AUCs with a very considerable R squared, about 0.25. And the p-values were absolutely very close to zero. And when we blasted this through publications, that the studies that have been conducted on these genes, we found that these genes were indeed related to the molecular and signaling pathways in invasive breast cancer, and their higher or lower expression indicated either the promoting of an activity that relates to the cancer or even promoting metastasis at some point. Further, we did radioproteomics experiment. So the aim one was to predict the estrogen and progesterone receptor statuses on these tumors, which are actually analyzed through the tissue slides post biopsy in the histopathology domain, and then further model. So basically predict these statuses from radiology image features, and then combine these statuses with the radiology features to depict protein expressions predictions in breast tumors. The way we did this is we took the very famous TCGA breast phenotype research group data sets with 37 radiomic features, which were highly curated by radiologists from top institutes in USA, Memorial Sloan Cancer Center, Mayo Clinic, etc. Pathologists reported the immunohistochemistry outcomes, the ER and PR statuses, estrogen and progesterone receptor statuses. HER2 we were not being able to use because majority of the samples didn't have that available, and we had a very small cohort to start with, but very confident cohort, I would say, because they were repeatedly curated by radiologists to make sure that we are indeed putting in very highly curated imaging features, and these range from texture, intensity, shape, etc. We splitted our data set into train test 95% and validation 5%, and basically splitted the train test for the train set into 80-20 of train test. The data normalization was set to scalar, and all these could be set automatically through ImaGene, such that you don't have to spend time coding for these or like making a parameter file every time. The classifier we used was random forest, which performed the best. Again, this was tested through a lot of classifiers, like support vectors, MLP, etc. But in the end, we found that random forest did work best for these. The threefold cross-validation score obtained was 0.24, and as you could see that it correlates sort of with the mean square error. If you take the root of these, it would land closer to your cross-validation score. That means that you're not overfitting the data. And then the AUC of ER status was obtained for test set. It was 0.96. For validation, it was 1.0, which makes sense because ER is more prognostic compared to PR. If you pull out any publication that would say that estrogen receptors are very prognostic of outcomes in breast cancer, like how would a patient perform on therapy, etc. So we then basically, so the conclusion was, yeah, we were able to predict ER better than PR, but then we said that let's mix up the radiology image features with the predicted ER. Again, this is a predicted ER at a very high accuracy, to see if it would predict the protein expressions in these tumors. The data sets were split as, again, now that we combine these data sets, 85, 10, 5. Models were used regression based now because protein expressions are more continuous compared to binary with respect to ER or PR statuses. So ER statuses are zeros and ones. Radiology are more quantifiable features. Protein expressions are also quantifiable, but they are again in the continuous form. So you have to use like regression-based models. And the specialty of multitask models is one prediction learns from the other. So let's say there are a couple of imaging features, texture, shape, intensity, predicting a particular protein. The model would continuously learn from these predictions and see if other if other proteins could be predicted along with the same protein, and that could be reflected in the results that we got. We got the top 3 proteins as p-cadherin, EGFR, and p4b, where p-cadherin performed mid-level expression and p4b performed at high expression levels, and EGFR at lower expression levels. And if you could pull out the publications, you would see that p-cadherin's low to high expression regulate epithelial to mesenchymal transition, a very famous EMT transition, which leads to metastasis of the cells to different organs. So they get converted from epithelial to mesenchymal type and float around in the blood and gets metastasized to other organ, let's say lung or brain. So the fact that we are getting p-cadherin at the mid-level here does suggest that these samples could be in their transition phase, not transition to metastatic yet, but could be between primary to metastatic. And we did, when we took these samples, they were claimed to be primary tumors, but you never know what's going inside them, right, with the biology of those until you detect these proteins. Again, these proteins were measured using a very famous reverse protein array. It's called RPPA. It's very famous. It was done by MD Anderson Cancer Center. So very highly trusted expressions. Further, we got low expression of EGFR, and EGFR are known to disrupt p-cadherin. So the fact that we are getting low expression of EGFR does say that may have played a role in boosting p-cadherin's expression. And then INPP44B protein is known to promote the WNT-cadherin signaling in breast cancer, and beta-cadherin and p-cadherin were found to be highly co-expressed in breast cancer, which reflects here that INPP44B was also high, which may have played a role in boosting the p-cadherin. Again, these are the studies created by the machine learning models, but takes us one step closer to the multi-omic research, where we could now validate this with functional studies and see if what we are seeing is actually correct. Peak gatherings are also known to classify the high-grade and low-grade histological subtypes of cancer, and we are working on this to kind of include that as one more feature type in our model. Further, there was a different data set for the invasive breast cancer from Duke University that we used, where we tried to use the imaging features from MRI, but they were from different machines, so the normalization was not achieved until then. Something that we are also working on our end. But what we were able to use was clinicopathological parameters for pre-therapy and post-therapy. So we use the pathologic response to neoadjuvant therapy that is given to the patient either as radiation or chemotherapy. We studied what was the tumor size, node staging, tumor staging, pre and post therapy, and then saw if that would allow us to predict distant recurrences. And we were able to go up to the AUC of 0.98 to predict whether this patient would recur with a metastatic disease after 60 or 90 days of follow-up. And we are validating it with more patients at Dartmouth. So this also speaks to the cross portability of the AI models trained on one Unicity data set and then validated on that on the different Unicity data set, which is very needed in today's AI world, where you want to cross-validate your models between the two institutes or more than two institutes globally, so that you could achieve some truthness of the model globally, and then perhaps take it to the clinical world, of course, through the rigorous validation. Conclusion is ImaGene is first kind of GUI-based software that allows you to work with plethora of parameters at your fingertips and conduct ML AI modeling, choosing the model types. Please go to the ImaGene's webpage and try to log in. It's a free account to create, and you can start experimenting there. Or there's also a GitHub page for command line interface users. Of course, you need some experience to work with ImaGene, like how it would yield the results and reports. But please feel free to contact me through LinkedIn or some other modes like emails so that we could set up a collaboration and sort of work towards a consensus models for cancer therapy and precision medicine. Thank you, and I would like to thank my PIs and co-PIs since these were the slides from my PhD study. There's more that I could have presented today from Dartmouth-Hitchcock, but I think the time would not allow. But I'm happy to talk to you anytime you wish to. Thank you.
Thank you, Shrey. If there are questions, or do you want me to